So in these notes, we're going to cover protein synthesis, which is the process where you make protein. And this is kind of what we've been talking about for a while now. The ultimate goal of transcription and translation is to do this, protein synthesis, to actually build a protein. So we talked about how DNA goes to mRNA. That is a process called transcription. So DNA to mRNA is transcription. And then you take your mRNA and you send it out into the cytoplasm where it joins up with a ribosome and some tRNAs and you build a protein. The process of reading the mRNA in sets of 3 and the tRNA knows, which amino acid to bring in to build the protein. That's what we're going to decode in these notes. So how does the tRNA know which amino acids to bring in? So this is a DNA code. So notice how you have lots of C's, T's, A's, and G's. It's pretty repetitive. Sometimes you have lots of letters in a row, etc. This is an RNA code, so it is the same, except it doesn't have T. Instead, you replace it with U. It also can be fairly repetitive, just like DNA. So a codon is a set of 3 letters that designates one amino acid. So that's 3 nitrogenous bases. So ACU, that's a codon, or GGG, that's a codon. Any 3 set of nitrogenous bases. And an amino acid may have more than one codon. So AAA, AAU, AAC may all code for the exact same thing, even though they're 3 different codons. There are only 20 amino acids in your body, but there are 64 possible triplet codons. So tRNAs, 64 possible tRNAs out there with their anticodons that match that, because that's what you get when you say, what are all the possible combinations of A, G, C, and U in sets of 3? And you come out with 64 possibilities. But you only have 20 amino acids, so that's why there's repetition there. And some codons tell the ribosome to start or stop. So you have 1 start codon, that's methionine, and then you have 3 stop codons. They just tell the ribosome to go away. Here is a codon chart that we're going to be using. You'll see a couple different versions of a codon chart. This is my favorite one. It's in this wheel shape. It is much easier to use, I think, than the square ones, but I'll show you how to use those later this week. So in using this codon chart, you start in the center and you go out. So an example of AUG, the way I would use this is I would start in the center here with A, and then I'm in this quadrant here. And I don't leave this quadrant here. This is the A quadrant. And then I move to the next circle, and the next letter is U. That's right here. And then I move to the next circle here in this segment, and then the letter is G. So A, U, G. And so that codes for methionine. So you start in the center, and each letter you just walk outward. So A, U, G codes for methionine. Okay. So follow this path. So what about GGG? So here you have G, 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 you get glycine. GGG codes for glycine. All right, I'll let you try this one. Give you a couple seconds to read the chart yourself, and then I'll tell you what the answer is. UCA. Good. Hopefully with your eyes, you went over here to serine. UCA, serine. All right, try this one. Good. Hopefully with your eyes, you ended up down here in histidine. CAU, histidine. Try this one. Hopefully you ended up over here with alanine. So GCA is alanine. But so is GCU, GCC, GCG. Those all code for the same thing. They all code for alanine. How about UAA? All right, hopefully you ended up at stop. Now, stop is not an amino acid. Stop is a stop codon. So UAA just tells the ribosome that that is where that protein synthesis is going to stop. So remember complementary basing, so on DNA, A is T and C is G, but in RNA, A is U and C is G. So the U gets replaced by A, or the U replaces the T in RNA. So DNA says ATT. Well, I can't look up ATT on this chart. There is no T's. So I have to transcribe this into RNA. So A goes with U, T goes with A, T goes with A. So what I'm actually looking for is UAA. And so UAA codes for stop. So how about this one? Do the transcribe and translate in your head, and we'll see if we end up in the same place. So DNA says CCG. What does the mRNA say? And then what is the amino acid? All right, CCG pairs with GGC, GGC, and you end up at glycine. All right, try this one. What's the mRNA? What's the amino acid? Do that in your head, and we'll see if we end up in the right place. All right, if the DNA says CTT, then the mRNA is going to say GAA. So GAA is glutamic acid. How many different ways can you make phenylalanine? So if we look here on the chart, there is phenylalanine right here. And phenylalanine has UUU codes for phenylalanine, but so does UUC. So there's two different ways to make the amino acid of phenylalanine. How many different ways can you make methionine? Methionine is right here, and there's only one way to make the methionine. It's always AUG, and that's because methionine is a start codon. So you only need one way to say start. How many different ways can you make serine? So serine is up here with UCU, UCC, UCA, and UCG. There's 4, but serine is also over here on this side of the chart. And so AGC also makes serine, and so does AGU. So there's actually 6 different ways there to make serine. So notice that the amino acids are sometimes located on different sides of the chart. So the end product is protein. You're trying to make what's called the primary structure of a protein. This is just amino acids, amino acid 1, amino acid 2, amino acid 3, amino acid 4, all joined together, and these white stick bonds are called peptide bonds, all the way through hundreds of amino acids in a chain. And this is what the process of translation will get you. And then later on, that amino acid chain, or that polypeptide, will go off to be folded into its right orientation. So it builds a protein that actually functions inside your body. And that is the process of protein synthesis.